Great. It's good to see some friendly faces here. All right. Welcome to the final session of the International Data Week 2023. What a great marathon we've been having. Thank you for joining us at this last session. This is a birds of a feather. We're going to be exploring how we describe samples of various different types across different disciplines of study. And so we have a series of 8 different speakers coming from different perspectives on this challenge, and then we're hoping to follow up with some discussion topic areas on that. The idea for this Birds of a Feather actually originated out of the World Fair Project. I'm sure everyone now has heard of the World Fair Project. That it's coordinated by CODATA and the Research Data Alliance with 11 different case studies across different disciplines, several of whom are part of this session. And this idea came out of a lot of the work that we've been doing in that project. Basically, I think it's pretty clear, samples are taken in every field of study, but there's so many variables around what those samples are and how they're taken and the various dependencies that are important to capture there, not only for the people who are sampling it in the time, but also anybody who wants to reuse the samples themselves or the data from the measurements around those samples. So there's a lot of needs around this that it's pretty complex. And so this is the kind of topics that we're hoping to think about. And as I said, we have 8 different speakers who will be speaking briefly to various challenges that they have with that. From this series of talks and the discussion today, we are hoping to kind of start compiling a little bit more in a formal way the range of these needs, especially around things like types and origin and workflows, both for collecting samples, but also processing samples and other requirements so that we can start seeing how we can sort these and organize them and maybe formalize some of them. Also like to identify the existing classifications and identifiers and other technologies that these disciplines are using currently and have been working on to try to support their work. We're looking for harmony. We're looking for opportunities to coalesce. It may or may not lead to the possibility of a working group in RDA if we're at a point where we can be producing something functional, or it may be that we have a little longer way to go. But we'll definitely be learning from this discussion today. So I thank everyone for participating. Just a few of the very many resources that actually are available currently in terms of identifiers and semantic description that have been used and we can be helping with some of these challenges. Some are used by different disciplines more or less successfully. Some are not being used in some disciplines at all. So I think it's an opportunity to understand what works really well, what's missing, how we can connect more dots around utilization of shared services. And finally, before I hand it over to the speakers, I did just want to acknowledge the co-organizers and speakers in this series. It's several people coming from the Physical Samples Interest Group in RDA. We have colleagues from the Photons and Neutrons Interest Group, the Chemistry Research Data Interest Group, the Improving Global Agricultural Data Community of Practice, and those are all RDA groups. And then we have several different projects from the World Fair, several different work packages, chemistry, nanomaterials, geochemistry, and agricultural biodiversity. So this is a really nice spread of colleagues here today. And without further ado, this is our agenda. Here's the link to the shared notes. As you're listening here, if you're also prompted to think of existing things that you're using or questions that you're having when you're trying to work with data or either reuse data and or share data and or curate data, particularly around samples and sample provenance, please jot them in the notes so that we can start collecting the range of challenges that we are all sharing here. All right, and so that's our discussion. I feel like we have a lot of repeat slides here. So I'm just going to launch into the first talk. Will be Kirsten Elgar, sorry. Thank you. She's from the Physical Samples Interest Group. They've done a lot of work on the International Generic Sample Number. And you've spoken about this several times this conference, but thank you for bringing this here as well. Thank you.
Yeah, thanks for that. I'm actually standing here on behalf of Kerstin Lennart, who has to leave, but we are both closely related with IGSN and the International Generic Sample Number, and the overall motivation is definitely to make samples fair. IGSN originated from the observation that it is really hard to track research results across publications if they derive from one sample, for example, from a marine cruise, and they're now distributed to many, many laboratories, and each rename them, of course, according to their local conventions. And then they publish under the name, and then you might have data from the same rock or subset of a rock, but you never know that how they can combine, or the need for having unambiguous sample names. So for that, the IGSN, the International Generic Sample Number, was invented. But the overall task is that samples have to be easily discoverable, open accessible, documented with sufficient information to make them reusable. That is the FAR, I would say. When the samples are linked to the data derived from them, they become interoperable, especially the results, and to the interpretation on these data published in literature. We can put everything in DataCite metadata or in other metadata, so there are means that is nothing new. But therefore, the precondition is that samples need to be globally unique. No, they need to be assigned with globally unique persistent identifiers that link to persistent machine-readable metadata. And that is the purpose of the IGSN, the International Generic Sample Number, that we have recently renamed from International Geosample Number, after we discovered that samples are not only relevant in the geosciences, but far beyond. So now we have changed to be adopted. That's exactly that globally unique and persistent identifier. It is guaranteed to be unique via a central control mechanism. Until 2022, we used the Handle server. Now we partnered with DataCite. We use DOIs. And these IDs that are actually glued or sticked, and you see these QR codes on samples, they link to the online representation or sample description. They can even be used if samples are destroyed during an analysis, because then you can still have the full record of the sample and say, this is what I have done, and this is the reason why it has been destroyed, because of a specific analysis method. IGSNEV is the governing institution. Well, it's a non-for-profit organization that has implemented the global registration mechanism. We were founded ourselves in 2011, already using DataCite's model for architecture and governance. Since our partnership with DataCite, we can ensure the technical sustainability and scalability of the IGSN PID. So we have the IGSNEV still to manage and grow the community around the IGSN, and also to serve as central information hub. But we use the technical implementation of DataCite, and for that we have we have mapped the core metadata from IGSN to DataCite. In practice, it meant that from January on, any IGSN can only be registered with a DataCite DOI. They recommend to use a separate repository, calling it IGSN IDs, resource type physical object. It also means that each DataCite member or consortium member can assign DOI without even getting in contact to IGSNEV. But this would be, of course, our wish to be in contact, because we might have experiences that we can share with you and ensure that there's a consistency. We have an initial mapping from IGSN metadata that I will show a bit more in detail in the last slide, and to the DataCite mandatory fields with some few additional fields. But still rely on our rich sample metadata that are accessible on landing pages at the registration agencies. And in the first part of the year, we had to re-register all more than 10 million IGSNs to DataCite DOIs, and you can find them. I just want to, because we are speaking about metadata, that is just in the model of the original IGSN metadata. It's meant to be modular with a very small number of mandatory fields that we used for the handle registration. And then we had the most, like we call it, common kernel, which is we're supposed to be agreed among the allocating agent metadata that are that allow to build a central catalog, for example. And then because samples are so discipline specific, we were very open for allocating agent to develop their specific add-on metadata. And this model is, of course, highly appropriate for samples and can never be mapped to the data side things. So I think that's the challenge, but that's still the point of discussion. And I think I've spoken longer than I'm supposed to anyway. Thank you. There's one more slide, but I skip it. I skip this and then we go to the next one. Oh, no, goodness. Okay, we're back here.
Thank you very much, Kirsten, for setting the stage and sharing with us your experience so far using the sample ID. And I think that slide you showed with the different metadata pieces and then what's currently mapped to today's site is kind of insightful. So we kind of have a picture of where the challenges lie. All right, next up is Deborah Decker. Deborah, can you hear us?
Yes, I can. Can you hear me?
Absolutely. Why don't you just go ahead and get started? You will need to tell us to advance your slides for you. Go ahead and get started.
So you're going to present my slides through there? Yeah. I cannot see them in my screen. Okay.
You can't see them at all in the Zoom?
Now I can. Thank you very much, Leah. It's great to be here with all of you. So my name is Deborah Drucker. I am a data manager at Embrapa. Embrapa is the Brazilian Agricultural Research Corporation. It's the largest organization on agricultural research in Brazil. We are almost 9,000 employees working all over the country, and agriculture is a very important topic in Brazil. So we do a lot of research, and I'm also currently co-chairing the IGAD Community of Practice, and I'm the lead of the case study on agricultural biodiversity of the World Fair project. So in agricultural sciences, we collect a wide variety of samples, biological ones, so we collect plant and animal or microorganisms, and we also collect environmental samples, such as soil or air, and from that we extract a wide variety of data, from genomics to chemical concentrations. So instead of considering this is a lightning talk, and instead of trying to talk about all the variety that we have, I chose to bring this particular example on soil samples, because this is I have been recently contacted by this community to work with them on the data part. So more specifically, I'm talking about this project called See Around. It's aiming on synthesized findings from a worldwide consortium of long-term agricultural field experiments, and it's focusing on soil organic matter sequestration and persistence. So it's focused on carbon sequestration and reducing greenhouse gas emission. So I forgot to ask you to oh, thank you. So we collect, it's challenging because this long time research experiments, they are conducted worldwide, and they differ very much on the crop and on the type of soil. And as they are long-term, and some of them are decades, or even in England, we have more than century experiments like that. They vary a little bit on the depth of the soil and on the methods themselves, and it's really challenging to harmonize the data from such a variety of different approaches that are going in parallel for such a long time. If we could go to the next one, please. So one very important point is the sampling design. So this is from a publication from FAO that's focusing on providing guidelines for measuring soil carbon stocks and stock exchange, carbon stock changes in agricultural areas. So we need to think that when we are sampling soil, we only have a dot in a large area. So it's important to consider that we need to have the information about the sampling design to understand how the carbon estimates are made. So this figure is here in this FAO guide, and I think it shows a little bit how challenging it is because it can vary very much. It can vary very much how the sampling design is done, and this is also something that it's a contextual information that needs to be expressed in the data itself. Next slides, please. So what the community brought to me is this spreadsheet with 19 tabs. There are some tabs that are more to express, to give guidelines how to use it, but I was really stunned when I saw this spreadsheet that's being used all around. It is EJP soil template. EJP stands for European Joint Program, so it's also being used in a variety of initiatives in Europe, and it's being also adopted in other contexts. So all the important information of the experiment is supposed to be expressed here, and also the metadata, the authors and things like that, and the data itself. And it includes the treatments, the land use history of that particular area, and the number of replications that the experiment have. And it's all supposed to be inserted in this very colorful template. Next slide, please. So I just brought this as an insight, and this is my last slide. It's probably the same for other samples too. We need to have the information about the experimental design. In this particular example of soil, the layers where the measurements has been made is also important, because people do measurements in different depths, and it also needs to be harmonized. As I mentioned, the treatments and replicates need to be also expressed, and it's also challenging. And I think it's important, this example of the spreadsheet, because that's something that happens a lot. So that's what the community is doing right now, asking the researchers to fill those very complex spreadsheets and not machine friendly ones, and I think we can do better than that. So I think that this will help for our discussion, and I'm looking forward to it. So I just will ask you, Leah, to show the last slide with my contact, if anybody would like to know more about this particular example, and I'll be here until the end of the session to discuss. Thank you very much.
Fabulous. Thank you. Thank you, Deborah. All right, Montserrat. So Montserrat will give us another example in microplastics. Go ahead and introduce yourself.
Yes, I do. Can I move this slide
the big green
myself?
arrow goes forward. Yes, you can.
Okay. Hello, everybody. My name is Montserrat Filey. I work at the University of Geneva. I'm not a data scientist at all. I'm a chemist, so I produce data. But at the same time, I've been putting other people's data together for 20 or 25 years. So you said this big green arrow at the top. This one. Now I'm going just to show you an example about nanoplastics in surface waters, as you probably know. And plastics in general is a big problem nowadays, pollution problem, and this is due to the fact that we are producing since the 50s a huge amount of plastics. But this is not the main problem. The main problem is that most of these plastics are discarded. So these plastics are not recycled. These plastics are not eliminated from the system. So one of the results is the presence of so-called microplastics, which are small pieces. The definition is variable. Not everybody agrees on that, but let's say pieces that are less than 5-10 millimeters in size. And this can have two different origins. One is that we use them as such. We produce them as small ones, like in cosmetics, but most of them come from the degradation, breaking of the big plastic pieces. Now, this research field has been producing a huge amount of papers, as you can see at the right-hand side. Essentially, not because it's a very big problem, but because it's very easy to study that, even if you have no knowledge on chemistry, no knowledge on colloids, no knowledge on anything, but you have a microscope and you get money. So this has produced a huge amount of plastic data, most of it a very low quality one. One of them, not all of it is low quality, but a lot of it is low quality. Now, what we are interested here is not in judging the quality of the data, but in considering that all this data is lost in practice. All this information is lost because it's not deposited anywhere. And even when people say that there is a sharing of the data, most of the time it's just, okay, you ask me and I will give it to you. So in fact, it's not a real data sharing. So just to situate a bit of the problem, what people do is go to some surface water, would be the ocean, could be a lake, and then they take the sample by using some kind of mantatrol or some kind of device that essentially filters the water. And then they get this kind of stuff. And this can later be treated very often by using all kinds of different procedures because, as you can imagine, you get also algae, you get all kind of stuff at the same time as your plastics. And this can later be size fractionated or not. And then you just count and measure. And so this is a very special problem that is not just for plastics, it's also for natural colloids. But the problem is that you need to know the size of something that is not regular, has not a regular size. You need to count the amount of these. And then if you are rich enough, then you also look for the type, so for the polymer type. So you have plenty of different problems together, let's say. So what I've been trying to do is to put some order or some logic in all these, collaboration with Leah and with Stuart. And we have been trying to figure out what is the information that is interesting and is needed in order to ask people to create some kind of metadata template, or could create a metadata template that also helps people to give all the necessary information. Because now what happens is that most of the people just give partial information. There is no real regular system. So some of the information is the usual one, like geographic location, sample identification. So it's common with other type of samples. Then you have the methods, which is this huge variety of them, because there are kind of treatments. And then particularly when people try to identify the polymers, they can use FTIR or they can use Raman with the associated libraries. So there is a whole big stuff behind that. It's even the microplastic definition, because as I said, it's less than 5 millimeters for some people, less than 10 millimeters for some people, and the lower size, it's also undefined. We have problems with units. because you have people expressing the results at number of particles per square kilometer, which is kind of strange for you probably, but this is related to the sampling stuff. So they just move these sampling devices on the surface of the ocean or the surface of the lake for some time. So this means some kilometers. So then they relate what they measure to the kilometers, square kilometers, because it's the distance times the width of the device. Other people at the same time measure the volume of water that has been filtered and then express that in number of particles per volume. Others weight instead of counting, and then you have mass. And some people move from mass to number, which implies quite a lot of hypotheses regarding size, regarding density. Then there is the object definition. And there I just went through one of the journals that where these people usually published. And I just looked for a year, so last year, and I found all these different types. So people say, okay, I've seen so many fibers, so many fragments. There is really no common numbers for that, names for that. And it also has a dependency on the origin. For example, all Chinese people who are very active now, they all the time say that they find where it's the thing, but it's not micro lines. They find lines all the time. Nobody knows what maybe they know, but we don't know what a line is. So probably it's a fiber for us. And other people, usually British people find a lot of threads instead of fibers. Okay, so even there is this kind of uncertainty. Then you have the polymer definition, polymer type, and there is, people say it's PET or it's polyethylene or whatever. More than 50 different types in 1 year. Some are the same, but with a different name, no identifier at all, as you know. And then there is the problem of particle size distribution, because some people just make fractions, and the size of the range of these fractions is completely variable. And sometimes people also make the distribution of the polymers a function of size. So you have all kinds of combinations of this information. Another information that very often people give, which is a very particular one, is the color. And then you have some people find color, others find colorless, which I don't know what it means, but it means because it's different from white transparent, which is around there. So you have all kinds of possible qualifiers. Now, the point here is to discuss whether we should fix some kind of specific types, or we should allow people to use any qualifier and then try to put them together later. This, I just would like to say that all this is important, not just because of academic, in academic context. We are now in the discussions of a new United Nations treatment, which will be legally binding concerning the plastic pollution in the oceans. And for that, these people, which I'm involved with them in some way, they are saying all the time that we need data. They need data. This data exists. Sample has been gathered somewhere. Sample is not available. It's no way to find this data. So thank you.
Excellent, Montserrat. Thank you. Thank you for being willing to share your your adventures through the microplastics literature. Okay. Next, we have online Alex Prent. Alex has been working on the Work Package 5 Geochemistry in the World Fair project. And I know Alex has some ideas about how we can start with the research object in a very essential way. And Alex, I'm just going to ask if you could spin through this pretty quickly so we can keep on time. And you just need to ask us to advance your slides. Can you hear us? Alex?
Try this. Can you hear me now?
Yes, we can.
Seems to work. Okay, great. Thanks. Yeah, so Alexander Prent, I must say I'm being supported by Oscope and CoData through World Fair Project. And this presentation is really about an idea that I got from spending time organizing this session. And it's about the starting point, or can we find a core research object or globally unique persistent resolvable identifier that notes the start of research. So next slide. Yeah, so we observe things, we observe not just things, but we can do experiments, interviews, surveys. We can observe people's interactions, for example. When we do research, it starts somewhere, and it doesn't have to be physical. Next slide. To point to those observations, we come up with an idea or touch Cupri, and we have all kinds of solutions there. But next slide. If we can, this is usually a physical thing or piece of data, and they're domain specific, these solutions we come up with. So couldn't we just find something, the starting point, I put it there in a modular way that's more universal and reaches across science disciplines and just notes the research starting point, only that. So go to the next slide. I guess for me, that would be the simplest terms, time and place. You know, if you can make a identifier out of that, it would pretty much be automatically unique, because who's going to do an observation at the same moment in time, the same second, and at the same place you're at? Nobody's going to do that, because the next slide, this example, if I were to make an observation somewhere 10 kilometers north of you guys in a garden, grab a leaf from a plant, it would pretty much resolve in this identifier over there. So the 10th of October, the 26th of October, about this time, 10 minutes ago, and at that place. Now, if you go to the next slide and you'd note those latitude and longitude in about 10 meters of precision, you'd result in a 120 square meter area. You can see all the people around you. Maximum 120 people could stand there, right? And it's very unlikely they will be doing the same sampling as you in the same second. So how you do that? Next slide, you just grab your phone and you take a picture. It has all the information you need. So maybe that's making the conversation a bit more difficult, but I just wanted to throw that in there as an idea I came up with. So thanks, everyone. Let's have a chat about it.
Thank you, Alex. So, yep. Thank you for drawing out some important facets of sampling for us. Next, we're going to move to another in-person. We're kind of juggling here. So this is Isla Lynch. She's going to be talking to us about the nanomaterials case study in World Fair. Thanks.
Thank you. Good. And the green button is there. Yes. Okay. What do I need to do? Just press the green button. There we go. So I pulled together a snapshot of some of our sampling challenges. So distinguishing between the sample as the object and the sampling as the event or a process or process. So yeah, just this is part of what we've been thinking about in the World Fair Work Package 4 on nanomaterials. So nanomaterials, the starting point is an intentionality. So unlike maybe the microplastics, which were something that is a pollution, or more like the air pollution that were the diesel exhaust particles that we've a long history of learning about. With nanomaterials, we're usually intentionally making them, but we are not that good at controlling them. So we end up with huge distributions of, things like particle size or the distribution of where the surface modifications is. So this is just an example of sort of different particles and their particle size distributions. So we can characterize them pretty well. And we can know quite a bit about them. And then, I don't know what the other one was trying to show. I can't remember what I was trying. Oh yeah, just to sort of show that it's not just size. We also then have differences in zeta potential. And when we try and do batches, so this was a specific study where we were trying to keep things really controlled and see how much variability we would have between them. So here we were either keeping the temperature the same or the starting concentrations the same and so on, and then characterizing them. And you see, like, even though in the groups that were made the same way, there's quite a bit of difference among the particles in terms of their physical characteristics. So how do we then capture that within and between sample distributions, and do they matter? Do we care? But what we've been thinking about, and this was work that was done in the CoData working group on universal description system for nanomaterials. And there they had the idea that we can think about an individual particle as a nano object, and we can describe a whole set of characteristics about that individual particle, but that would be on a single particle. Whereas actually the samples that we're dealing with in real life are collections of nano objects. So we have a slightly different set of properties about them because maybe a distribution, we have surface distributions, topology, and so on. So we may end up with a different set of identifiers. And actually, one of the things we've been working on in terms of our extension of the INCHI to nanomaterials is also that it's not going to describe an individual nanoparticle, it'll be this collection of nanoparticles. So that's one of our challenges that I just wanted to throw out there. The other one then is this, and I can't believe, when I dug out this paper, I was going, oh my goodness, we did this so many years ago and we are still not reporting properly. Things as simple as the batch number and the day it was made and the day it was opened, because the minute you open, you might make your particle and you might have it nicely under argon or whatever to keep it perfect. Then you open it to do an experiment on it. And from then, well, even in the jar, it was aging. And certainly the minute you open it and start doing things to it, it's aging, it's transforming. So this is just an example of the same material with different coatings and different times and, different storage conditions and how it aged. So you can see sometimes they turn black, it means they've oxidized, sometimes they precipitate, all sorts of things. And then you do the toxicity on them, maybe being aware that they've changed from when you first got them, maybe not. So, we know this at some level in our community, but that knowledge doesn't necessarily translocate. And we're certainly not reporting it sufficiently. And then the last one, just coming back to the intentionality then, we do a lot of tests where we're intentionally putting them into the Daphnia. I can't do a presentation without my beloved water fleas, the Daphnia. So again, here we have, is our object of interest a nanomaterial or the Daphnia or both? In actual fact, we're interested in both what's happening, what the Daphnia are doing to the particle and what the particles are doing to the Daphnia, and how we think about all of those as well and how we document and describe. So again, just wanted to throw out, that we're in both cases, we might have more than one object of interest and how we capture that. So just some key considerations to throw into our discussion: the sample complexity within and between batches, the sample evolution, both during storage, on dispersion, when we add it to an environment, and the intentionality that, here we are purposely making things or purposely adding them to systems to look at what happens both ways. Super, thank you.
That piece of objects for us. Okay. All right, next is me, actually. All right, thanks, Isolt, for that very complex use case. All right, so just going to talk a little bit about some of the issues in chemistry. There we go. It's finally advancing. And this is coming from an IUPAC perspective. IUPAC is International Union of Pure and Applied Chemistry. It defines many standards about naming chemicals and chemical methods of analysis and other concepts in chemistry that are reused in a lot of other disciplines. And I think that's really kind of what we're thinking about here is the summary of a lot of the challenges across these is the chemical composition is an aspect of what a lot of other research areas need to know. It's not the only thing they want to know about their sample, but it's most often an aspect of it. And so what do we need to do to think about how chemical composition can be a part of the sample provenance, for example. And once you have that in your provenance information about a sample, for example, anyone coming along to reuse the sample and or the data coming from it is able to tie into a lot of other data that's already been reported and analyzed around those same chemicals. For example, solubility. If you've observed some chemicals in your sample and you can get a sense from the solubility how readily those chemicals are known to disperse in different environments, which could be useful to know, like nanomaterials. Right? Challenge that we have though is there's a lot of different ways of assessing and expressing composition and the concentration of chemical species in a sample in any given material, partially depending on the physical state, also depending on the conditions under which the sample was analyzed. So there's just a lot of variety, and we have the same challenge everyone else does. I mean, there's a lot of standards defined in chemistry, but not necessarily consistently used, or and not necessarily easy to convert from those different types of reporting. So I kind of pretty much already said this. One thing to add, though, is that a lot of the chemical data that's available and the chemical standards that are defined are thinking about single chemicals, and that's a very useful way to think about chemicals and to to analyze them and predict on them. But in fact, actually, all samples in the real world are mixtures of chemicals. You may not be able to ascertain more than one if you have a small impurity, but there are, it's like what I was just saying, the moment you open the bottle, there's impurities happening in there. And these are also dynamic systems. A lot of materials are very sensitive to the environment and the conditions of the environment, just like Isel was just saying. So, you have to think about, are you studying the overall system properties, so you can capture chemical at a point in time that's useful to know, or are you actually studying the changes over that time, the kinetics? And so if you're studying a fixed point in time or a representative description of the chemical composition of a sample, you need to maybe wait for equilibration to happen. So there's a lot of complexities in taking a chemical composition as well. Just want to pop this in here. So one of the biggest challenges that we have is notating what chemicals are associated with something. In the chemical informatics field, we have a lot of different ways that we've developed to do this, because we like to reuse this information in a lot of creative ways. The known chemical space is hundreds of millions, and you can project out to billions of chemical molecular entities. And so there's a lot of need to be very detailed in how these are described. But then it puts up a lot of choices in front of the community. Which one of these approaches do you use to describe your information? I've circled the INCHI down at the right-hand side. This notation is machine readable, it's algorithmically generated, and it was developed to provide a canonical representation of a chemical, a molecular entity, for that purpose of sort of registering it in databases and being able to match it across different data sources. So it is a useful component to thinking about it. And so, for example, another project that we're working on IUPAC is notating mixtures. As I said, you have multiple components in a mixture. They have different quantities being in the mixture. Some of them might have been in a mixture first, and then something else was added, and that order and that hierarchy of those ingredients and their relationship to each other is important as well. It will change, could change, the behavior of the material. So it's important to notate all of these things. So this is why we're trying to work on a notation that captures that. And the notation leverages the INCHI of the components and then builds some notation to express the relationship between those. And in this way, people can use that INCHI to link back to other information about those components, but also understand the relationship of them. On the right side of the screen here, I have inchy, and this approach assumes that you can specify exactly what chemical entity you have. It's not always easy to do that based on what you're able to assess or what someone else has notated and you're reusing their data. You often don't necessarily have that much information. It might be a natural product that hasn't been fully characterized. It might just be a real incomplete description that you found. So we're also trying to work on how you can still use a lot of information from those more ambiguous descriptions, and what is useful that we could still use from those. So that's an ongoing project. Go, go, go. Sorry about the quality of this, and then I'll stop talking. We also compile data from the literature and evaluate it because we understand that these chemical properties, such as solubility that I mentioned, are really useful for other people to know. And that involves, again, we have to compile from the literature. We have to make sure that we can harmonize together the components that different people were studying, the study conditions that they used, the conditions of the measurement over the time of the measurement was taken, the forms of those components. So there's a lot of information that we need to actually compile data. So we are a reuse case here. And then once we've managed to compile that data, we can evaluate the quality of the information in it. So that assessment of quality is about the science there. And then we can publish these, and these become reference data. So as others who are measuring information about chemicals can use that as a benchmark. But to get that benchmark data, we need to collect original measurements. And some projects that we have going on this, you have to throw out as much as maybe a third of the data that's been reported because it's not adequately described. And so that's all I have. Hopefully that wasn't too long. Next we have Rolf. Thank you. Come on up. Rolf is coming to us from the Photons and Neutrons Interest Group. Go ahead.
Yeah.
Big green button.
Okay, yeah. As she said, I'm Rolf Koll. I'm from the Helmholtz-Zentrum Berlin für Material und Energie, from the photon and neutron science. And just one slide from our background. So our community presents large-scale facilities such as synchrotron light source or free electron lasers or neutron sources. So large facilities. And most of us are user facilities. That means we provide the possibility for scientists to submit a proposal in order to apply for instrument time. And that one important thing to notice in that context is, so we are not, say, representing a particular scientific field, but rather a class of experimental techniques. And that means we have users from basically all scientific disciplines coming to our facilities and performing experiments at our facilities, which makes, of course, some things a little bit more complicated because we have a large diversity of samples that coming to our facilities. And of course, in order to make the data that we are measuring fair, of course, we need, say, an accurate description of the sample that has been put in the beam. So just to give an idea of what we are facing, we have all sorts of different examples, starting from relatively simple things like single crystals, crystal powders, functional materials, to more complex structures like solar cells, batteries, or all things that are composed together. We have macromolecular crystals, that means proteins. You have seen the spike protein of the COVID virus. The fact that we know how it looks like is due to measurements at synchrotron light sources. But we have all sorts of other things. We have archaeological artifacts. Nebra sky disk was once in our facility. We have fossils. We have artworks like paintings that are investigated with tomography. We have living plants. I could add to that. We have organs of animals and all sorts of things. And that means there's little hope to, say, come to a comprehensive metadata standard that would fully describe that sample that we have. But as an alternative approach, so there's one thing that works, say, in the positive direction for us, that many of our samples are created in the lab specifically for the experiment. And so the approach that we can take is, if we, of course, all these preparation steps, sample characterization, all of this is, of course, very documented, creates scientific artifacts like lab notebooks, data sets, and so on. And if you link all that together, then we come to more or less a perfect sample description. Because if we, say, record the history of the sample, then we know what it is. And so rather than putting too much effort in, say, working on sample description standards, we rather would want to rely on sample PIDs in order to combine the information that we already have more or less at hand. And of course, we also have samples that are not prepared in our labs, but then often they come from some external collection, like these artworks or other things. And then, of course, we would need to point to the entry in that collection, hoping that some curated sample description is then there. So that was so basically our strategy, but still there is something that could be done that would be very helpful for us, is, say, some semantic artifact, some vocabulary or ontology or whatever, to do the top level classification, just to put things in one drawer to distinguish the archaeological artifact from the single crystal. So that level of abstraction, that would already very helpful. It would need to be able to cover all samples that we have. And basically that means all samples that appears in the science. But it only needs to be, say, could stay at a very coarse level, because once we have some characterization, then we could apply a domain-specific sample description. Yeah, and that was already my last slide. That was the statement that I wanted to make.
Thank you, Rolf. I really appreciate having that facility perspective to covering across a lot of disciplines. That's kind of a nice foil. And last but not least, we have Arifan Gregory. Arifan is co-leading the work package 2 in the World Fair project on domain interoperability. The green arrow.
I'm familiar with this equipment, sadly.
Yeah, I figured.
So my name is Arifan Gregory. Simon is sitting there, looking at a smartphone. Together, we are part of the center of that pedal diagram. So work package 2 is the cross-domain work package in World Fair. And so we're concerned really with the interchange of data and metadata across domain boundaries. And domains can be a lot of different things, but that includes infrastructures. And I think from some of what you've seen today, there's some relevance there. And we recently had a workshop at Schloss Dagstuhl. And some of the people here were present at that. And I'm going to talk a little bit about that, because we put on the table the question, could you have a generic description of samples and events and other kinds of things that are actually studied, like directly observed and measured? And we weren't sure the answer was yes to that. And I think you've heard some different viewpoints about that today, and I'm going to disagree and agree with all of them. The key thing that we're working on is called the cross-domain interoperability framework. And what we're looking at is a core set of metadata that can be used to exchange data and fair resources, so metadata, across domain boundaries. And yes, we're talking about one size fits all, because this isn't a model so much as a meta model. It's a set of standards that are by their nature cross-domain, used in coordination to facilitate most of the kinds of functionality that people want when they're doing FAIR at a level appropriate to cross-domain use. Now, that was a lot of noise I just made. It's really complicated to create a standard that allows fair sharing of things ubiquitously, but that's what we're trying to do. We don't think it's impossible, but again, we're relying very heavily on the use of meta models as opposed to models. And most people who think standards think models and ontologies. That's not really the way that this works. The idea ultimately is to have this core sort of lingua franca so that domains can map their things to it and then get some visibility across the holdings in other domains and be able to identify them, use them, and maybe learn more from the domain fair standards about those things. I'm not going to go into massive detail here, but the idea is some things have to be domain specific and some things don't. When you talk about the structures of data, the structure of metadata, those things don't have to be domain specific, but can be described independently. Now, at the Dagstuhl workshop, we put some questions on the whiteboard. And I'm not sure I was completely happy with the answer, but I think I was happy with what came out of the workshop. We said, what is this data about? How do you answer that question when you're asking it of anything that you might discover anywhere in the world? Like, that's a big question to ask. I've already seen the feature of interest phrase used, and that causes me to see red a little bit, and I'll talk about that. But your questions, what is this data about? What is it? Are events and samples and other, and I'm going to use a phrase that everyone hates, units of study, be described in a generic way in relation to data? People talk about features of interest. I have a big background in social science. People aren't features of interest. They get really insulted when you call them features for some reason. And I'm glad you don't do the pan stuff on people, by the way. Plants, that's okay, but people, no. But it's really, you get some really, really different perspectives in different sciences, as we know. And how do concepts, the sorts of things that ontologists define within domains, and variables, the sorts of things that are contained in data records, how do those things relate to units of study? How do we understand those relationships? Now, this is a pretty brave new world. A lot of people in standards assume the implicit knowledge within their domain that informs their recording of data. And that's not always true, but it's very often true. And when you start looking at cross-domain data exchange, you can't do that anymore. People don't understand the implicit things within a domain because they're not members of that domain, of that discipline. And what do I need to know about an experiment and about the process in order to really reuse data effectively? So we came up with a couple of different things. There were a couple of different activities. And the first of these, we tried to jam this all in one slide and it didn't fit, so I made two, or Simon made two. You may be familiar with GBIF from the sort of biodiversity world. They came up with a unified model, and they identified sort of methodology based on PROV and OGC's observations and measurements spec. And so we took that and sort of, Simon was actually the chair of this, but took a bunch of different domains and created these kinds of diagrams. This is carrying on from Deborah's soil thing. I think this was Deborah's example, right? And I'm not going to go through this in detail, but the point is that you have samples and events and activities, all of which are necessary information in understanding what the data actually is. And this was an exercise in identifying more specifically what those things are. What do I need to record about the context of the data, of the observation, in order to actually responsibly and accurately reuse it? And again, this is something that's sort of maybe not fully baked yet, but that is tending in that direction. I think there's some value in using this approach. But what came out of this was a lot of information about how we think about the things that are measured and what else we need to know. And a big part of doing a sort of cross-domain standard is saying, what is the process and provenance information? What is the information about the experiment, the study that you need to know in order to use the data? And that's what this methodology was getting at. And then things got much worse last Sunday because we hit people with this. Okay, now this is a meta model. When I talk about features of interest in like an O&M sense, I'm really talking about what we call the unit cascade. Apologies to people who think that means unit of measure. It does not. It means unit of study, okay? A unit of study is the thing being observed and measured. And there are a lot of different ways to think about it. Now, Alexander talked about time and geography. We recognize in this model, for instance, and you're looking at this model is taken from DDI CDI, which is not maybe super important here, but when we identify a population, that is a set of samples that are being observed, those things, and they may be individually, are specific to a time and a place. And we do recognize that that is a special thing, but they also have a set of characteristics here termed a universe, which is a bunch of things of a particular type that are distinguished according to some set of characteristics. Are you confused yet? Because it's going to get worse. The unit type, which is a word everybody hates. Michelle knows all of this because she worked with DDI for a long time. But unit type is the kind of thing being studied. Is it a person? Is it a nanoparticle? It could be one of the huge numbers of vocabularies describing the unitary things that are studied. And those things have types. A unit is one instance of one thing of a certain type being studied. All of this can be viewed through a conceptual lens. Now, I've just done what we call the unit cascade from the bottom up. That is answering the question, what is this data about? When you look down the other side, what we call the variable cascade, that is answering the question, what is this data? So we have this idea of a concept, which is like age or height or viscosity. It could be almost anything. But the concept is applied to a unit as a characteristic. And when we have that, we call it a conceptual variable. It's a certain property about a certain kind of thing. And then you say, well, how am I going to represent that? You get it to units of measure and these kinds of things. And you add that information at that level. And then you have specific instances of those measurements represented in that way of that particular thing. And the instance variable can be understood as sort of a column in a table, for instance. It's a set of like measurements of a very specific type. And this is the kind of meta model you have to get into when you start creating standards that are describing data ubiquitously and sort of universally. Now, this sounds a lot like philosophy class or something, and in some ways it is, but if you apply this correctly, I think you can get to a place where you have these models that you can begin to search across everything and find what you're looking for. Unlike feature of interest, right, which could be pretty much bloody anything, this actually starts to nail down the particular gradations within that become important when you're trying to apply descriptions to things like that. All of this presumes the kind of identification that people have been talking about here, by the way. So I don't want to hit you with too much, because this is pretty much this is a pretty crazy model, but it's been road tested a lot, and I think you'll see the outcomes from this might be pretty good. We really want to continue pushing this forward and seeing what comes out of C. diff. I think in terms of samples and events and those kinds of measurable things, there is goodness here to be applied to the kinds of work that's being suggested in other presentations here. I think this is important work. Within World Fair, we'll continue doing this for a while, and we'll see what comes out of that. I think there has to be some continuation of that work, and I don't know what that looks like. I guess that's what we're going to be talking about now. But we wanted to present this just because I think it's kind of an important approach to the subject from a very different perspective, which is how do we transfer this information across domains when sometimes the best description is within the domains. So.
Thank you. Awesome. Great. Thank you. Ending on that really meta level out there. And I just want to mention that the discussion that we had in Dagestall that Arifan mentioned, and following up on Sunday here at this conference, we're pulling together a paper that can provide some of those examples, and those examples are drawing from a lot of the domains that you heard speak today. So this paper we're hoping to get out early in the new year, I believe, or some kind of report at least to be available. Oh, but that's the draft. That's not external, right? So watch for that. Before we get into some follow-up questions that I or others might have, I wanted to open it to the floor. Are there people who have questions about what you've seen, or you have been working in this area and want to contribute some experiences? There, okay, he's right there. Oh, go ahead first. Yeah, I see you, Barbara.
Thank you. I'll go first. I was interested to see that you put up a thermodynamic phase diagram. And I mean, this is a really big problem. So, I mean, do you think the whole phase diagram is a property of the sample? Or, I mean, let's say if you've got a a piece of ice and you warm it up and melt it? Does it become a different sample on it? How do we handle that? I mean, it's especially difficult when you've got really subtle changes like different magnetic structures in a material or something like that. Are they all different samples?
Yeah, that's a really good point. So, you know, this is the phase diagram that it was mentioned here, and you know that you have temperature and pressure and the physical state of the material. You know, well, this is for water. You know, phase diagrams are usually done by chemical. So, you know, and then it changes phase, and I guess that's part of what I probably didn't say very coherently is, you know, either you're taking you're sort of doing a snapshot in time and location that has to, you know, at a certain temperature and pressure and composition that gives you whatever you're looking at. But then, you know, any amount of change, no matter how subtle, over time and location could have an impact on this. And Isolt mentioned in the nanomaterials use case, and Kirsten, who's part of the geochemistry project, also has mentioned it. You know, on the other hand, you may be wanting to study that change over time. And so then what do? You put a lot of time sequences in place, and then you can assemble a diagram that looks like this. So yeah, there's a facet of the time and location here, and there's a facet of the conditions. I don't know if that's quite what you were getting at, but yeah, it's very dependent. Go ahead.
Kamil Jubek, University of Vienna. As a follow-up to your question, I completely disagree of calling it a dynamic phase diagram. There is no such a thing like a dynamic phase diagram. There is only one phase diagram that gives the energetic minimum for given pressure and temperature and composition. And what is else we can call a pressure temperature diagram, but not the phase diagram, because phases are actually defined in physics. I wrote a paper on that. I'm glad to share it with you later on the definition of a phase diagram quite recently.
Yeah, that's a really good point. So what that is, that's the definition of the phase diagram there. I wanted to emphasize this puts parameters around what gets represented this way. Now, this does make the assumption if it's, you know, of composition, like this would be pure water, right? And in the real world, you never have pure water. So these become more reference benchmarks rather than, you know, the real world.
Sorry, if you just allow a remark, it's not what I understood as the point of the question, but rather the generalization also to what Isel presented for biological samples, like a piece of tissue or even a live animal or live plant. So there is a concept of event, and also in this overall generic model, there is a concept of event that is related or belongs to a protocol. I don't know whether it's optional or whether it's compulsory, but coming again from maybe biased, we are dealing with some fish samples and sea organism samples. They tend to decay and they tend to change and live or rather die on their own. And there are events that are not comprised by the protocols. So I would probably concentrate more of these events and try maybe to put them into some context or into some, whether they do change the property of the sample insofar that it becomes a different thing. So that's what I understood as the essence of the question.
Thank you.
Not really whether it's called phase diagram or dynamic diagram.
Yeah. That's a really good point. Yeah. You know, so I think it does depend on how we define things. Yeah, because that's what's going to make the boundaries between sample one, sample two, time one, time two. Barbara.
So sorry. So I would perhaps like to dive a little bit more into the value proposition of having a unified theory of all samples, because I mean, I go back to my favorite toy model when I think of things like this, right? I think about Wikipedia, and you know, if you look at a Wikipedia entry for a person, historic or alive or dead, you know, it follows a certain schema. You're comfortable with that. You could look at a historic event. You could search for a place, a country. You could search for a food product. I think we intuitively look at a Wikipedia page and we don't really have a lot of trouble understanding that these are different things. So but clearly we're all in this room because we want some satisfaction out of having a unified theory of all samples. So help me here.
Thank you. Go ahead, Barbara, and then we have another question up here.
One of the slides of our fund, bringing together PROVO and observation measurement, is that the result of the second week in DAX tool? And I missed somehow the property, the observer property of O&M, and I would like to explore a little bit more in detail this. Is this published? Can I step into this? Because I think we are really investigating how to combine O&M, iAdopt, and and the prof stuff.
Over the last 2 years, I think, GBIF has been involved in a process to create their unified model. The driver for that was the realization that in the biodiversity world, there was suddenly a whole load of new data types and data sources. And their current model was getting unbearably stretched, couldn't accommodate these new forms of data. So they have been going through a process of trying to understand the data types that come from their various communities. So to do that, they have used a methodology which combines more accurately SOSA SSN and PROVO to do this modeling. Mm-hmm. So each of these, the purple things is an event, each of the yellow ovoids is an entity, et cetera, et cetera. And they've adapted it to their needs as well. So I wouldn't get hung up on how strictly it conforms to observations and measurements, et cetera. That's kind of not the point here. The point is that they've developed an approach to working with their communities to try and understand the set of processes, the steps that go through the creation of the data. And we found that very interesting when Tim Robertson showed us this in the Darkstall workshop, because it helps us do something very similar within the World Fair project. And what we take out of this is, okay, one, the enlightenment of understanding the processes, which is useful in any case, but most importantly, what are the key steps that need to be recorded, and what provenance information do you need from those in order to understand the data? And then that combines with, the next slide and this. And to answer the question about which referenced Wikipedia and why we're doing this, I mean, I don't think it's just, from a World Fair perspective, it's not just an intellectual exercise. We're not trying to model samples to have one unified model sample, which will make us feel intellectually happy and warm, but also lead to a whole load of endless arguments and, et cetera. The reason we're trying to do it is to identify key connections between data sets for interoperability. Those include and are not limited to those pieces of provenance information and the unitime conceptual variable, etc. Arfan wants to qualify what I've said.
I do, and I'm in no way disagreeing with what Simon said. This is not about a unified model of samples. It's about a unified model of the description of the glorious diversity of samples. Okay, I said meta a couple of times. It's about unifying the description, because I don't think there is a single unified model of samples. I just don't. So, and maybe I'm wrong. Maybe there is, and you can show me wrong. I'll be happy. But that's.
Sorry, I give it. But you know that iAdopt is also a meta model of a unified description of variables. So
yeah,
and I wanted to see how.
Great. There was a gentleman up here first. Could we? He's been waiting, so I wanted to thank you for your patience.
It was kind of in response to the thing at the back, which I agree with. The comment from the back there, and it's kind of exactly what Rolf was saying. I'm from the same community as well, for the samples cover more or less anything we can fit inside the hutch. You know, it's vast numbers of different things. We'll do nanoparticles, we'll do proton crystallography, we'll do fossils, we'll do anything you like. And it's not about a universal description of the samples, it's about finding a link so we can get a description of the sample. So so we're not, you know, we don't want something that covers everything. We want something that we can index and find a description. And then that domain will have to understand that description. I'm kind of slightly worried that some of this stuff is going into almost like a universal model of capturing the whole of research.
Mm-hmm. Yeah,
and
it
you're
definitely is.
Synthesis, you're capturing observation, you're capturing methods. And it can get very, very large. And if you've got a working group description here with a piece of work you want to do that's something useful, I think we need to focus on something useful.
Yeah, I think that's a very point is very well taken. I think for the workshop scenario where several of us just came from it, we did find it helpful hearing our whole stories just to get a feel of the variety. But that I think feeds into this idea that we need to surface up that what needs to be in that meta indexing layer, what needs to really be up there so that we can get down into the domain, because clearly reusers need to get down into the domain. That's needed. That was what we learned, but we need to also figure out how to navigate that up to the meta level so you can find stuff. So I think it's what? How do you find the links back to, the more specific domain description? Okay, so I want to hear more from you guys, so go ahead.
No problem. I wanted to add another line of thoughts here. I see a fundamental difference between provenance in data or metadata and provenance of real samples. Yeah, just everyone to be aware there's a fundamental difference, which also affects reproducibility and trust in a provenance chain. There's potentially a certain an uncertainty factor in the provenance chain. You never know if you recorded all the events that acted upon a sample. For example, was it light exposed? Was it warmed up over a certain time, or exposed to a certain gas and reacted? All these kind of things can be unknown in the provenance chain, but have a real effect on the sample. And that's the difference we need to be aware of when comparing provenance chains between data and metadata, where you can very well reproduce the data operations that you did between one and the other version of data or metadata.
Yeah, thank you. That's a good point. Yeah, go ahead, Isolde. Is it easier to come up here?
So you're absolutely right. We spent a lot of time talking about, and for example, in the geology example from Kirsten, where, the sample is collected and it's, under a certain way, it is transported under known conditions to the lab. So, in some cases we know because it's never really out of our control, but that you're right, all of that steps need to be documented. And the other case study that hasn't come up that I think was relevant was we also had, museum collections. So, and there again, well, Beth, do you want to add something on that since it's yours, and just, the stuff about how you manage everything and know everything about what has happened, but you keep it as metadata and keep it internal and don't necessarily share it with the researchers? Sorry to throw you on the spot.
I knew there was a roving mic. Yeah, no, thanks for including me in this. I feel like a bit of an interloper. Felt like a bit of an interloper in Dogstool as well, because I'm coming from the cultural heritage domain. But of course, sometimes your samples wind up in the cultural heritage domain and they're managed by museums. And we do have systems by which we track what happens to things while they're in our collections. What we discovered is that we're actually not very good at sharing that information. We think of it as kind of administrative data. But as we were talking through, research processes, how you would use things in chemistry, how to use things in what happens in a lab in nanomaterials, we talked about what happens in a museum and what happens when people come and approach us about collections that have been in museums, and what we do or do not tell them about the number of conservation reports that might have been made on a particular object, or whether it's been sent and exhibited somewhere and what the light damage was during the exhibition process. We do often, I mean, not all museums, because not all museums are capable of keeping very extensive records, but in a lot of cases we do have that information and we do record it. And so how do we share that?
I just want to make a remark on that at that point. The perfect is the enemy of the good. I mean, of course, there are lots of things happening to the samples that we do not control. But still, it is very helpful to record everything that we do have under control and to document that.
Yeah, thank you for that. I did want to just quickly say, offer the online people. If you have a question, please let us know. Someone's monitoring the chat. Okay, go ahead, Kristen.
Yeah, I think adding to what Rolf just said, this is also a known issue with IGSN.
Yeah.
A normal use case is that they're included in repositories, in core repositories. So everyone, like there's the hierarchy of like a drilling project, you have the whole, you have the core sections, the samples. So if I go to a repository, I collect a sample, this comes within IGSN, but then I go to my lab in another university and I make subsections, and following this hierarchy of IGSN, each subsample should have a new IGSN. But would the university know about them? Would they have the possibility of assigning IGSN? And at this point, and I've talked to several repository managers, they usually say this is the natural end of this provenance chain. And I think this is also something that still requires a lot of awareness raising, because as soon as we leave the data management domain, the researchers don't care. They just, unless they are in a project where they are supposed to have it. So it's getting better, but there's still a lot of work to do, I would say.
Yeah, moving from space to space, right? Go ahead.
So in a slide you showed on solubility,
okay.
You actually had three fields for description: conditions, experimental data, and estimated errors.
Yeah.
And estimated errors, or reporting estimated errors, are very much discipline dependent, because in crystallography, everyone is reporting estimated errors. In chemistry, it's like half to half, and there are some disciplines that people just forget about it.
Yep.
And it's also a source of final uncertainty of your information on the sample. And in case of course, of the fate of the sample, what's going on, was it irradiated or not? There are a lot of different conditions, so we don't know the outcome. But in case of errors, if only we can save them as the data or metadata items, we should do that. And we should make people, experimentalists, aware of that, sensitive to this problem.
Thank you for calling out the importance of uncertainty. That's certainly definitely important in chemistry and in other fields. That the uncertainty that might come from the instrument, from the conditions, and that's part of what we do in IUPAC is we're looking for that, so that as we combine data, what we do is we actually not only evaluate all the data that we've gotten out of the literature, we evaluate how much they know about how precise that data is, and that's why we do that, because it is a very talk about ambiguous space. Yep. Go ahead, Simon, and go ahead.
This relates a little bit to what was just said, but I want to sort of pick up on that. Of course, we all agree with Voltaire, but I want to sort of respond and clarify that what we're trying to do in World Fair and CDIF is not, aiming for, this modeling at that sort of level of perfection. But what it is aiming for is, we might say that perfection or is the enemy of the good. But if you don't have, we're responding to some very specific case studies and use cases within those case studies about data reuse and above all data combination. And if you don't have sufficient metadata, you simply can't do it. So while we might say the perfect is the enemy of the good, insufficient information means that you can't do anything. And I think this is, this is also relevant to what was just said about uncertainty. It's hard, but we do need to try and understand the uncertainty in each and every data set that we think we might need to use. Otherwise, what are we doing? And if we're combining data sets from different sources, we do need a method and a strict method for how we also combine those uncertainty levels. And the statistics that relate with that. Otherwise, again, what are we trying to achieve?
The method of
We've just crunched a number of databases together, but we don't really have any science anymore.
Thank you for emphasizing that, Simon. I think there's an online thing, and then I want to kind of summarize some things. You want to just read it since you can see it?
Yeah, it's a comment here from Abraham. Digital specimen model is an example of cultural heritage specimen, and it's got a link through to something from Disco. In cultural heritage, specimens can be stored in multiple sites. Each piece may be considered an independent sample, but some provenance is needed to link them together. And I guess it's that theme of linking coming through again. And then Abraham's background is chemistry. Various samples from a material may be characterized using different techniques. The papers tend to publish a mix of results from different techniques. And then that's that case of teasing apart all the things that get mashed up at the moment into sort of one great big report.
Yeah, and actually, probably the papers people publish are almost like a reuse case of the data, right? So if you're using a lot of different techniques, that's really actually a bunch of different data sets being combined there. So that they could just notate their metadata a little better, we would all be better off. Thank you for those comments. I just want to note that we're almost at the end of this session. I think when we planned this, we hadn't yet had the coming together conversations at Dajtal within the World Fair project. So it was a really opportune actually that when we actually arrived here, we'd had that conversation just 2 weeks ago. So it was great to be able to share back with you what we've been thinking about, but also hear more thoughts coming from all of you. But what I guess what we've learned from both Dajtal and here is, where maybe we're not ready to crystallize around things, but I think one thing we'd like to do is we have our paper. We'd like to share that out shortly that we're putting together. But I think figuring out how to follow up on some of these threads of what's a meta model. And I think another thing that we want to learn more is how well some of the existing resources have been used. So I was going to pick on Kirsten a little bit more right here. If you have a moment to reflect on how well the IGSN is working, for example, that might help us think about that. Did you have one more thing you wanted to say since you're holding the microphone?
Very shortly.
Yeah, that's fine.
So moving from this good versus the perfect, or the degrees of reliability of some information on the samples, and then again drawing parallels between there is the museums and cultural heritage, there are also parallels with the industry and the value chain and the supply chain and traceability and trust. And there is a dreadful buzzword looming here and I will mention it now, the blockchain. Because when you're recording this trace of events, there are some that are probably if you're really sure that something happened, then there is this method of ledger of immutable change that you practically introduce to the data set. So just a question, because it was never mentioned by now, do you think that it is a sort of still a sort of overkill or not yet useful for this tracing of events, or would it maybe have some relevance here?
So I will just note that IUPAC has a study going on looking at that question of blockchain in different use cases in chemistry. And Stuart's online and has been part of that project. I don't know if we have time now to comment on it, but I think I did want to call out, you mentioned industry, and definitely in our field, you cannot share your data unless you can really, trace it at that level. And certainly with internally to companies, they have to track their data at that level. So there's definitely an existing culture in some areas of the industrial sector where they're tracking pretty much as much of everything as they possibly can. You know, I don't know, Stuart, if you want to just mention real quickly what you've learned in the blockchain paper relative to how useful is it or not?
I just wanted
I probably can't really speak to that at this point, only to say that the actual paper itself will be out early next year, or the white paper will be out early next year. I'm not sure that I'm qualified to make any comment on that.
Okay, yeah, it's looked at a lot of different use cases for how you could use blockchain in different areas, different fields of chemistry. So we'll make sure that gets out to the community because it follows changes in information. So it's a model there that we could think about as a mechanism. Is there any other? Go ahead. And then Kirsten, behind you, sorry, first. No, she's already got the microphone. Go ahead.
Hello. Hi. So I'm not a researcher at all, and this is not my domain at all. But I'm sorry. Yeah, I'm a designer, and I work for an ELN that has a sample management system. And one project we did was look at a collaboration with DataCite, actually, where we implemented the very basic workflow for minting IGSNs. So say you collect some samples, you put them in the inventory system, then you can mint, the researchers can mint the IGSN, populate some metadata and publish it, and without having to deal with the kind of having to think about the infrastructure behind it. And it was actually, it sounded like a really easy project at first. It's like, well, it's just a basic workflow, right? You just kind of click on a button, but there was so many layers of having to learn the nuances of that workflow, both in terms of how do IGSNs work as an identifier and what do the researchers need? And then, like landing pages, like I kept having this conversation of, well, who hosts the landing pages? And it sounds like obvious once you actually know what you're doing, but I was thinking, well, DataCite, right? Because DataCite does everything else. They do the registration and so on, so they should host the landing pages. And the institution said, well, but we have already other landing pages for other things, so maybe we're supposed to host the landing pages. And so I think it's really maybe worth considering involving some of the makers of the tools that are used as well, because this metadata and the workflows will have to take place in some kind of structured digital tooling. And you can't support let's say all of the metadata fields from DataCite because most of them might not be used for a certain domain context. So it's useful to kind of find out a balance, I think, there as well. So I'd really love to hear from people about that.
What metadata and then how it's used and, how it can be leveraged. That's an excellent point. Thank you for that. Yes, so I've experienced a few different ELNs. You want to do one from chat, and then I was going to ask Kirsten.
So there's one from Stuart here, which sort of maybe moves us towards that possibility. Is there a working group here? But a working group on samples could do the following. What are the axes of metadata around samples? I think there've been lots of interesting comments about what is minimally necessary, what's out there already, and the outcome propose a general way to deal with samples, data, metadata, which perhaps recognize those bounds of possibility.
Yeah, I definitely want to acknowledge, I think we need to do more groundwork here and collect a lot more examples, a lot more input so that we can coalesce it. If the working group is the best mechanism for that, we could do that. Or if there's some other suggestion coming out of World Fair or CoData, I'd be happy to hear that. Did you want to just also, since I asked you, Kirsten, to kind of give us a little bit of lessons learned from over time?
Well, I think that coming to the first line of the slide, if we generally recommend the use of controlled fair vocabularies, and I mean those who are registered with SCOS RDF, then they are like, these would be the sources for ontologies. And if any group that is dealing with samples is trying to include these vocabs instead of dividing or like inventing their own lists or going away from anything, it's always easier to register your own vocab than to look what others have done and try to use it. But this could be an initial starting point for like machine to machine semantic ways of bringing samples together across domains, I think.
So kind of the combination of the PID and a high level shared ontology that references others yeah, something along those lines. Thank you.
I think that has been done in many life sciences and in the marine. So I think it's but this is the precondition. So we always have to say use vocabs, use the right ones, don't invent everything, and then it's going to be fine.
I think exploring what the parameters are around that will be really important. Go ahead, Arifan. Could you pass the microphone to Arifan, please?
I'd like to respond to that comment. And I'm thinking about this from the perspective of C. diff and the problems we're trying to solve. If this group were to create that kind of ontology, very high level ontology of the kinds of samples that exist, I think that would be an incredibly useful thing and would plug directly into the kinds of work that we're trying to do as well. So I would second that motion insofar as that's worth anything.
Excellent. I think it does kind of tie nicely into what you were saying, Rolf, about that kind of something at the high level, and rather than the grand model or the getting into the weeds, so to speak. All right. Barbara, you want to have the last word?
Yeah. Say something about blockchain and identifiers. Are you aware of decentralized identifiers from DeSci? They are working with blockchain technology. So might be interesting to look into that. And then I just wanted to say there are vocabularies about samples.
There are
among here with from the NERC vocabularies there are. So please make sure to connect.
Yes, an awareness and analysis aspect of the next steps is really important, I think. Yep. So I think we're over time, so I just want to acknowledge that you never have enough time to really get into the fun stuff. But given that there is sort of this paper coming out, and we are going to be distilling, I think, from the recordings, a lot of the great comments that have been made here, and maybe we could even work that into our report that's coming out of World Fair, and then we can from there consider the next steps. But I think watch this space in terms of another BoF session at RDA to see what bubbles up from that. And we'll be posting, I think, out to the interest groups that are involved in this project, that information as well. So, you're going to have the last word. Okay.
No, just as Arifan pointed out, the collaborative notes document, there's a place to put your name in if you haven't already.
Yes.
So if you haven't already, find a time to do it.
Yeah, excellent. Minimally your name if you want to be on the loop. And then like I said, we'll try to push this out through the interest groups as well as the World Fair channels. So thanks for your time and scratching the surface with us, I guess I'll say.